Okay, we are really lucky to have Dr. Anna Durbin here to talk about a specific broadly neutralizing antibody that is being used in a clinical trial. And just a few notes before we get started. So upcoming conferences on next week, we'll have a few cases and a little bit of an introduction to the HOT study, which is an ongoing trial recruiting through the ACTG for looking at menopausal therapy in women living with HIV. So hot flashes are actually the primary endpoint. So if you have patients who have hot flashes, please think about this study. We will have a break during August. And sorry, that's that thing that always happens. And then in September, the first week back, I believe, is an ID town hall, but then we'll have IAS updates on the 18th. The Polk lecture with Dr. Rochelle Walensky will be in September, and also we'll have Bartlett updates and cases coming up. So just keep those all in mind, and if anybody has anything they want discussed next week, please just send it to me, and we'll add it to the ones we have for next week. Okay, so great. So I'm just going to give a brief overview of neutralizing antibodies and broadly neutralizing antibodies in HIV, and then turn it over to Dr. Durbin. So neutralizing antibodies, so they are the little blue things in here that coat the surface of a virus and then prevent the virus from engaging its receptors on a cell so that it can no longer cause a productive infection. I think we were all became very familiar with these in the COVID epidemic when there were all those news infographics that had the spike protein and the antibodies. So what is the difference between a neutralizing antibody, one that binds and prevents entry, and a broadly neutralizing antibody? So specifically for HIV, that was developed through a number of different experiments where they looked across multiple isolates from patients, so 200 different clones, for example, isolated from different patients, and then tested the ability of antibodies to neutralize each one of those individual variants. And they then divided viruses into 3 tiers, although there's like 1A and 1B. The tier 1 are the easiest ones to neutralize, meaning most antibodies that are specific to HIV to that region will actually bind to it well and prevent infection. Then tier 2 gets a little bit harder. It's harder to find the antibodies that will neutralize it. And tier 3 is the hardest. So these are the viruses that it takes a very specific antibody in order to block it and prevent infection. And so now the goal of developing a vaccine would be to induce an antibody that could neutralize whatever virus came along. So you can imagine, since we're only looking at like 200 or so in this kind of a panel, that would have to be covered among those. So you can rate your antibody once you isolate an antibody from an individual based on what tier level it can neutralizing. So most people who have HIV will develop some what we call autologous neutralizing antibodies. So those are antibodies to their own virus, that if you took their own virus, the antibodies would bind efficiently to them and would help to prevent infection. But only a small number of people living with HIV develop some of these broadly neutralizing antibodies that can neutralize lots of different types of viruses. And there's been a lot of research in the field to identify where these broadly neutralizing antibodies bind, because they have to bind to part of HIV that does not change a lot. Otherwise, you will lose your neutralization capacity. And so this diagram here shows a number of the different sites that can be targeted by broadly neutralizing antibodies. Among them, the CD4 binding site here in yellow and the V3 glycan loop are 2 of the sites that broadly neutralizing antibodies have been identified from. And so this is from taking serum from people, isolating their HIV-specific antibodies, and then testing each antibody to see if they can find a single antibody that neutralizes a lot of viruses. So that's how a lot of these were found. These antibodies they require extensive, what we call somatic hypermutation. So that means lots of genetic changes in order to develop this broadly neutralizing capacity. And vaccination approaches have not come near to eliciting these types of responses so far. So there is now a strategy for trying to develop these. This is by identifying the very deep pink initial antibody that was the first one, and then sort of trying to graduate it along until you get to the very most mature broadly neutralizing antibody. And so people have taken a few of these very well-studied broadly neutralizing antibodies and tried to figure out exactly what sequence they went through to become broadly neutralizing, where they started from, and the different mutations that were required to get them. And then one idea is to try to develop a specific immunogen to use to get through each stage. So that if you actually took a person and gave them multiple vaccines over time, you would actually lead them down this pathway to developing a broadly neutralizing antibody. So that sounds great, except for if you need to have multiple different immunizations spread out over time, that is a very long-term strategy that would have lots of potential for falling through the cracks, not completing the series, not getting to the endpoint that you want. So it's not the most sort of compact and efficient vaccine strategy. So what are alternatives? So one of them that was very popular a number of years ago, or was thought to be a major potential, was a vectored vaccine. So this is when you take an adenovirus, you put in the bNAb that you want to make, and you infect the person, their muscle cells in some cases, and then that muscle cell starts to pump out the antibody that you wanted. So you have sort of your own little depot just producing the antibody over time. There's been some technical challenges of that, but it's not a totally dead idea. And then the other idea is passive administration. So either through infusion or subcutaneously giving these antibodies that we know to be broadly neutralizing to people. And that's what I'll just briefly talk about how has been used in some therapies. So for HIV prevention, there are the AMP trials done by the HIV Vaccine Network, HVTN, and HIV Prevention Trials Network. These were two different studies that used a CD4 binding site antibody, BRCA1. They had estimated that they thought 65 to 80% of the isolates from the trial sites would be sensitive to BRCA1. So every 8 weeks, they did an IV infusion for 20 months in 4,000 participants in US, Switzerland, and South America for a trial in cisgender men and transgender people, and in sub-Saharan Africa and cisgender women. They also offered PrEP, but that uptake of PrEP was very low. Less than 1% of the sub-Saharan African participants had therapeutic levels. So what were the results? So only 30% of the HIV that was acquired was actually sensitive to the antibody. Among those sensitive viruses, it had 75% protective efficacy, but that is not the result we want. If only 30% of the potentials you could get infected with are actually sensitive, it's not a wide-ranging strategy. So a single antibody is unlikely to be adequate, and there's lots of scaling and feasibility considerations, but this trial was really a heroic effort to try to look at this strategy. When we can't make it with a vaccine, can you just give it to people? And I think there's still a lot of thought about how that might be moved forward. So bNAbs for cure or remission has been another major focus. So the Rio trial is just an example. So this included 3 BNC117 and 101074-LS, both of them with a modification to make them last longer, in virally suppressed participants who had started antiretroviral therapy in an early stage of HIV. They interrupted their ART after receiving blinded infusions of either antibody or placebo and were followed for 20 weeks. If you were still suppressed at 20 or controlling at 20 weeks, you were offered another infusion. And their primary outcome was the time to viral rebound within that first 20 weeks. They defined viral rebound in a generous way. So it was the first of 6 values of greater than 1,000 copies. So if you went up and went back down, that didn't count as you losing control, or 2 greater than 100,000. So this is much higher than for an ART trial. This would be declared a virologic failure a lot earlier. And I'll get to why they did that in the next slide. 75% of those who received the antibodies in Arm A did not have viral rebound at 20 weeks versus 11% in Arm B. So it did show some efficacy. And then for those who continued and got the second infusion, 25% of Arm A and 11% of Arm B still had their definition of viral control at greater than 96 weeks. And importantly, at that time, all of the people who still had viral control did not have therapeutic levels of antibody. And so this gets to this idea that antibodies may be doing something other than just neutralizing the virus. And one of the ideas is that they may be having what is called a vaccinal effect. So if you look at this graphic, here's someone with HIV. They have their viral load rising in the mustard line, their immune response rising in the purple line. When you start ART, your viral load also comes down, but your immune response also decreases and remains kind of low because there's not much stimulation of that response. And so then if you stop ART, your viral load comes up and your immune response comes up, but it's lagging behind. And the idea is that with a bNAb administration, you might actually boost the immune response. And this is by making immune complexes that then more efficiently prime T cell responses, is one hypothesis at least. And so that your immune response rises up, even though your viral load doesn't come up. So if the viral load starts to come up, the immune response is more ready to do something to keep it down. So that's one hypothesis that's being explored in the remission and cure space. And then for treatment, the largest, most advanced study so far is on 2 antibodies. They are the same ones that I just mentioned in the Rio trial, but they now have fancy names. I can't say them, but they are TAB and ZAB. And they are to the CD4 binding site and to the V3 glycan loop. They have that LS modification for longer stability in the plasma. This was a randomized open label phase 2 trial in Australia, Canada, and the US that enrolled people with HIV less than 50 on ART with high susceptibility to these antibodies. And what they found, they were either randomized to receive lenacapavir plus TAB and ZAB or continue oral ART. And the primary endpoint was an HIV, actually it was HIV detectable greater than 50 at week 26. They enrolled 80 participants, and the results were 1 individual from the Len plus TAB and ZAB had an HIV greater than 50 at week 26, and none from the group that continued their baseline oral ART. So that's somewhat promising as a combination therapeutic. The one failure is high, but this is, again, one of the things that we've observed with injectable therapies is they're, unlike oral ART, perfect adherence doesn't always necessarily guarantee that you will have a maintained suppression. And then just in terms of will this work for the patients who we most might want it for, the Prestigo registry of individuals with documented 4-drug resistance assessed about 50 of the patients in their registry who had for susceptibility to these antibodies. And they found that in the 46 patients they were able to get results for, 76 were susceptible to TAB, 50% were susceptible to ZAB, and 41% were susceptible to both, which is what you would ideally want if you were going to use this as a therapeutic option. So it may, in fact, be something that we can think about for our patients with multi-drug resistance. So I would summarize bNAbs as potential uses across the spectrum of HIV prevention, treatment, and cure. Substantial questions remain, and studies are needed, which is the drum roll lead in to Dr. Durbin.
Thanks so much, Eileen. That was really a great introduction to what we're trying to do with our study here today. So, great. Let me move on. That was a great introduction. And what I'm going to talk to you about today is a broadly neutralizing monoclonal antibody whose parental clone was 118. And we're going to hit on some of the issues that Eileen was talking about in terms of sensitivity to the broadly neutralizing antibody and how it may be used not just in prevention, but treatment. And then I think what I would say is an aspirational goal of the company as well is for cure. So as Eileen mentioned, this antibody as well, oh, I just did something bad. Hang on, I've lost my, there we go. This has been modified to include a mutation that extends the half-life of the monoclonal antibody such that it can be given every 6 months. So if this were to be successful, patients would then just get an injection every 6 months to either prevent or control HIV. And I think the other, one other difference that this monoclonal antibody has is that it actually binds across 2 different sites. And I'll talk a little bit about that in a minute, meaning that it is more difficult to develop resistance to the monoclonal antibody because it requires multiple mutations to induce resistance. So it has a high affinity binding to the HIV envelope protein trimer. It does bind to the CD4 binding site, which Eileen mentioned is one of the targets of different broadly neutralizing antibodies. And it neutralized HIV strains irrespective of co-receptor tropism. And one of the, and I think Eileen touched on this a little bit, one of the perceived, I would say, benefits or of broadly neutralizing antibody is that they can directly eliminate HIV-infected cells and neutralize the circulating virus, leading to viral suppression. So it can be used as other sort of, other than neutralization, other antibody-mediated defenses to actually eliminate HIV-infected cells. This is being developed primarily as a therapeutic as opposed to a prophylactic, but potentially could be used as both. And why aren't we moving forward? Let's see. Let's see if I can get this. Let me go forward. Okay. So this was identified from a person living with HIV with elite neutralization activity. And they tested this as they were developing the product, testing the plasma, which neutralized 96% of a diverse 118 virus panel. So it was one of the broadest recorded, and I'll show you a slide in a minute that compares 118 with different broadly neutralizing monoclonal antibodies that have also been identified. So looking at different B cell sorting and single cell cloning, they isolated this antibody, and it emerged as one of the most potent and broadly neutralizing clones. It has a rare 6 amino acid insertion in the CDRH1, which allows it to contact both protomers of the HIV envelope trimer simultaneously. That has not been seen in other what we call VRCO1 class antibodies, which are the ones that have been studied to date most commonly. And because of this, it has broader epitope contacts and restricted viral escape options, so that it's much more difficult for the virus to escape neutralization from the antibody. So this is just looking here at 118, which is the antibody that we're talking about today, and other antibodies of the VRCO1 and other classes. Green means that that it's sensitive, that the virus is sensitive, and this is looking at different, looking at the concentration, the different mutations that have led to escape from other monoclonal antibodies. So you can see that the amount of antibody required to neutralize the virus in red means that the virus is resistant. It's greater than a 5-fold change in the ability, in the amount of antibody that's required to neutralize the virus. And throughout all of these different mutations, what can be seen is that the antibody retains its neutralization capability regardless of all of the different mutations here in the different sites. So this is just an overall summary when we look at this antibody compared with some of the other antibodies that have been studied, particularly the VRCO1 class, of which this is considered part of. It was out of a 208 virus panel, 97% of those viruses were neutralized. The IC50 is very low. If you look at it, it's the lowest IC50 of all of the different monoclonal antibodies, meaning it's the most potent, requires a much lower amount of antibody, which could be important in terms of how we deliver it. So the higher the potency, the easier the less volume of antibody required, which gives this the potential to look at sub-Q delivery or very small volume delivery of monoclonal antibodies, as opposed, for instance, to infusion of a large quantity. Now, I will say, when I talk about the trial, we are administering this as an infusion. We looked first in group A in people who were living without HIV, and we looked at a subcutaneous dose as well as IV infusion, and I'll talk a little bit about that in a minute. But what you can see is that 99% of the strains were neutralized at less than 1 microgram per ml. So it's very, very potent. It has broadly neutralizing antibody. It does target the conserved CD4 receptor binding site on the GP120 outer domain, which is critical for viral entry. Again, it has the 6 amino acid insertion, which was a somatic insertion, and as Eileen mentioned earlier, so that required multiple mutations during affinity maturation to reach this, but it contacts the adjacent GP120 protomer of the trimer, again, contacting 2 sites, meaning that it's more difficult to develop an escape mutation with this monoclonal antibody. And as I said, because of this, it makes it much more difficult to develop resistance to this monoclonal antibody. This is just going through some of the different VRCO1 class escape mutations. When they looked at the 118, which is the antibody we're talking about, it neutralized 7 out of 8 of the VRCO1 resistant strains with, again, a quite low IC50 range. The only strains that were able to escape were those with rare multi-site mutations, and that showed reduced 118 sensitivity. This suggests that this monoclonal antibody could be used in patients who fail other VRCO1 class antibody therapy. So they did map the escape mutations. And again, the reason that this is restricted is because of the multiple attachment sites or binding sites of the monoclonal antibody. So it may have more durable viral suppression. Those mutations, those viruses that did develop escape mutations, took a fitness hit with those mutations. So they had reduced replication capacity, even though they were resistant. So they did look at this monoclonal antibody in a humanized mouse model to look at how effective it might be. This was given as a single IP injection of 200 micrograms. They monitored viral load weekly for 4 weeks. All of the mice treated developed undetectable viral loads, so they had a complete virologic response. The viral loads dropped to undetectable within 1 to 2 weeks of a single antibody injection, and suppression was maintained through week 4 of observation without any rebound. In comparison, the untreated mice maintained high viremia throughout. So again, it induced highly effective viral control in this mouse model. And then they next asked, does this monoclonal antibody work against strains that are already resistant to clinical stage antibodies? So they looked at mice who were infected with the VRCO1 resistant HIV-1, the N279K mutant virus. This abrogates VRCO1 binding, but had no significant effect on the 118 binding because 118 does not critically depend on that contact. So the thought was that this monoclonal antibody could be useful in patients who were receiving another VRCO1 class antibody and who experienced viral rebound. They could potentially be treated with this monoclonal antibody. And again, what the purpose of the study that I'm going to talk about today, and I think what the vision of BioNTech, who's developing this monoclonal antibody, is that it could serve as a first-line therapy independent of VRCO1 class antibody exposure. So they're looking at this, again, as a therapeutic and also as a potential antibody that could be used in combination for a cure strategy. So the thought is they can pair this antibody with other VRCO1 class antibodies to prevent escape. They can, by understanding the CDRH1 insertion in interprotomer contacts, they may be able to design a vaccine. And ultimately, that is in fact what they're trying to do, is to use this study and to use this monoclonal antibody in combination with a vaccine as a cure strategy. Again, they also are looking that it could be used in PrEP as well because of its broadly neutralizing capability and the fact that it has the extended half-life so that it can be given only once every 6 months. So again, the other thing that they're looking at is a functional cure research. I think this is really in the future, but should things proceed well with this trial and additional trials, that is, again, one of the long-term goals of developing this antibody. So I think I'm going to go through this pretty quickly, but just in summary, I think it's a very strong candidate for clinical development as a long-acting HIV therapy, PrEP, and then in combination with other broadly neutralizing antibody regimens to really try to attain durable HIV remission with an easy schedule for people living with HIV. They could come in again every 6 months for injections and then be good to go. So we started looking at this monoclonal antibody along with another site in Germany. There were 2 sites that were evaluating the monoclonal as a first-in-human phase 1 clinical trial, looking at the safety, pharmacokinetics, and antiviral activity. of 118, which they've renamed BNT-351, looking at it in adults living without and with HIV. And Part A, as I said, was conducted here at Hopkins as well as in Germany. And this was conducted in people living without HIV. And we looked at 3 different dosing levels, 5 milligrams per kilo, less than 15 milligrams per kilo, and less than or equal to 45 milligrams per kilo. The 5 milligram per kilo dose was looked at both subcutaneously and as an infusion. They took the data. This monoclonal antibody was very well tolerated in all groups and all doses. The infusion was very well tolerated. There were not any infusion reactions. It's infused over about an hour. And so they have moved forward to look at evaluating this in people living with HIV. Originally, when they planned this study, they were only going to look at the less than or equal to 45 milligram per kilogram dose. They've opted to, based on the pharmacokinetics that they saw in Part A, look at also a 5 milligram per kilogram dose. Now, what I will say about that is the 5 milligram per kilogram dose that we're looking at can be looked at because we're only going to follow people on the monoclonal antibody through study day 56, because the 5 milligram per kilogram dose does maintain levels above those that are neutralizing through 2 months, but it doesn't maintain high enough levels through all 6 months. So how they're going to use those data to potentially try to extend the half-life or see if they can look at dosing between 5 milligrams and less than or equal to 45 milligrams per kilogram. The 45 milligram per kilogram dose had titers that were well above those that were neutralizing throughout the 6-month period of time in that Part A study. So again, they're going to look at only 16 people living with HIV, and you'll see that's because what we're really trying to evaluate this is in people who are newly diagnosed with HIV and have not been on therapy. Also looking at people living with HIV who have been on therapy but have been off of that therapy for several weeks. But the goal really was to see if they could find people newly diagnosed with HIV who have never been on ART. It's going to be conducted in Germany and then 4 sites in the US, only looking for a total of 16 participants. Again, I think that's because they recognize that it's going to be difficult to find 16 newly diagnosed people living with HIV. So this was Part A. I've just put the study design here. Part A, as I said, was done at 3 different dosing levels, 5 milligrams per kilogram, both subcutaneously and as an IV infusion, and then less than or equal to 15 or less than or equal to 45 milligrams per kilogram given as an IV infusion. In Part B, which is the part that we're going to be that we're currently recruiting for, is people living with HIV. 8 people in each cohort, the 5 milligram per kilogram IV and the less than or equal to 45 milligram per kilogram IV. They will get a single infusion, and then at day 56, we'll start combination ART therapy. Again, when they get the monoclonal antibody, that is the therapy that they're getting for treatment of HIV. They will start their standard prescribed combination ART at day 56 or earlier if they meet criteria to start early treatment, or if the participant wishes to start earlier, they can start combination antiretroviral therapy. So again, this is just looking at the different parts of the study. Part B is open label. There is no placebo control. Everybody will get either 5 milligram per kilogram dose or less than or equal to 45 milligram per kilogram dose. 8 people in each group. There are sentinel people in each dosing group. The way they've designed the study is that we can enroll there are 3 sentinels in B1 and 3 sentinels in B2. We can only enroll 1 sentinel a day for 3 days. And then once those 3 sentinels have been, the safety data has been reviewed, then we can enroll the remaining 5 volunteers in B1. And once the safety data for the 5 volunteers in B1 has been removed, then we start with 3 sentinels, again, just as I said before, in B2. This I don't have to tell you. Again, this is just, again, the sentinel review that I discussed. There's a 48-hour sentinel review after each of the 3 sentinels have been enrolled, and then we can go on and dose the remaining 5 participants. After IRC review of B1, then we can go ahead and start enrolling B2. Again, 1 participant per day until the 3 sentinels are enrolled, then a review after 48 hours, and then dosing the remaining 5. So we've been working very closely with Joyce, and Joyce said she's sorry she couldn't be here at the meeting today, but working very closely with Joyce and the Rapid Start program, as well as other HIV providers in Baltimore, trying to identify newly infected people living with HIV. We've been having weekly. Oh, thank you, Stuart. I appreciate that. I just saw your comment in the chat. We've been having weekly lunch and learns just to tell people about the study from around Baltimore, just making people aware that we're looking for people newly diagnosed with HIV. My staff and I are available to talk with anybody who may be interested at all. We can come wherever you are, talk with the potential participant and see if they're willing to enroll. I do want to say we're doing this very closely with HIV providers, such that even if a person is enrolled in the study, they are connected with an HIV provider at that time, and we're in discussion and sharing if the participant agrees, which I'm fairly certain they would. We would be sharing all of the different lab results. We get weekly viral loads during the study. Follow them very closely. We'd be sharing all of the lab results with the HIV provider, and really in close communication with the HIV provider to ensure that they're ready on day 56 to start combination ART therapy with their provider. Oh, let me come back here. So this is the primary objectives of this study that I put up, really looking at the safety, primarily the safety of the monoclonal antibody, as well as the ability of BNT-351, looking at the effects of that on viral kinetics in people living with HIV. This is just I don't really want to go through these in great detail. I'm happy to take any questions. The inclusion and exclusion criteria are relatively stringent in terms of what a person can have experienced in terms of PrEP, or if they had been on combination ART, they have to have discontinued it for at least 4 weeks. Again, there's other treatments that if they have been on, they would not be eligible. For instance, lenacapavir. They cannot have taken lenacapavir to be eligible for this study. They have to have a CD4 count greater than 400 and a plasma HIV RNA level between 1,000 and 100,000. So you can see it's relatively stringent. They're looking for people who have not advanced in their HIV. And again, they have to be willing to start combination ART at day 56 post-dosing of the monoclonal antibody, or earlier if they meet early start criteria. These are the criteria that have been set out in the protocol for when people can start combination ART earlier. And it has to do with viral load or CD4 count, non-virologic or virologic non-response, and then safety criteria. And then the bottom one is if they request to start combination ART prior to day 56, then they will be started. And this is another reason why we really want to work in close collaboration with the primary HIV provider caring for anyone who enrolls in the study. This is just some of the schedule, which I won't go through. We have handouts and everything that we can provide to anyone who's interested that goes through some of this in more detail. But, I did want to just stress that they are followed very closely. They get weekly measurements of their viral load, and they get every 2 weeks they get their CD4 count checked, and that has to do again with ensuring that we identify virological failure as quickly as possible. So these I've just put Megan, who's our study coordinator, and Sarah Sampson, who is our clinical director at the CIR, with emails and phone numbers. I'm available as well. We're really excited about this. I will say this is a little bit outside what my normal clinical trial wheelhouse is in terms of we've done mostly very early phase vaccine and challenge studies, but this has been a really interesting study for us to do. We're thrilled to do it in collaboration with Joyce and everybody at the Bartlett Clinic, and trying to, the CIR had a long history with Mary Lou Clements Mann of doing HIV vaccine studies. And we haven't been doing those since Clayton Harrow, unfortunately, passed away. But I think bNAbs are really exciting for the potential that they have in prophylaxis as well as treatment, and the potential even for cure. So we would love to work with anybody who has a patient who may be eligible for this study. Just reach out to us. We are easy to reach, can get in touch, and happy to see any potential participant at any time. And I'm happy to take any questions.
Well, thank you so much, Anna. And also, Laura can correct me if I'm wrong, but any participants who are interested, they can also call the CFAR hotline, which should be able to code. So if you as a provider can't keep, we'll put these slides, in a accessible place, but you can always, the CFAR hotline will always be available to connect people to any of our supported studies, which includes this one.
Oh, that's fantastic, Eileen. Thanks so much.
So any questions from the group, or Stu, do you want to nerd out about antibody CDR3 length?
No, I just think it's fascinating. One thing I was confused by was the notion that there's a 6 amino acid insertion and the motif given is 9 amino acids, but that's because germline has a different set of 3 amino acids in this CDRH1, which is weird. So for those who remember their med school immunology, this is a very strange insertion. Not supposed to happen.
I mean, for me, this raises one of the questions that I often have about this, which is that because some of these antibodies are so weird, will they emerge from just standard vaccination in the absence of the immune dysregulation that comes with chronic HIV?
Yeah.
Well, if I can provide a visual to underscore your point, I'll show you the map of these antibodies relative to germline. Just pulling up the share. Again, this is not what you came for, but I think it's kind of amazing. So 118 is the top one here. So what you're seeing on this top row of the colored part, this is 118, and here's the insertion relative to germline, and there's another table in this paper that shows that this is an IGH V1-4601 typed heavy chain. And so look at all the changes relative to germline. It's crazy how many changes there are. And then in the light chain, this is the light chain for 118 on the top. And there's a crazy number of changes in the light chain. So to Eileen's point, like when Justin has led work with Hep C, we never see this many changes in people with acute Hep C evolving to broadly neutralizing antibodies. HIV seems to pose a tremendous problem for broad neutralization relative to, for instance, Hep C. This is not usual to see this many changes in an antibody in a human.
Wow. Well, and I think it goes to Eileen's earlier point as well. I mean, even if you try to design, it would be interesting, to design a vaccine that can induce a similar, in theory, polyclonal response. Could you even do it? And I think that's a really tough question. I mean,
Well, particularly consistently, like you might do
yeah,
it occasionally, but to expect this to happen consistently, this degree of somatic hypermutation without untoward effects of having to hammer the host, I think that part is tough. I see Bob came off
Yeah.
camera mute, so I'd love to hear his comments on this.
Yeah, no, I think the amount of mutation is pretty astonishing. I have sort of a different question though. So Anna, essentially, as I understand it, essentially it's going to be monotherapy
yes.
in the beginning of the trial. So is there a reason to think that the results are going to be any different than monotherapy with any of the sort of conventional classes of antiretroviral drugs, which sort of always leads to a drop and then resistance?
Yeah, I think I can tell you their aspiration based on what has been described. Their thought is that one, it's extremely potent, number one, and two, it binds sort of to two of the different protomers. So their thought is that they will be able to control it. It'll be more difficult to have an escape mutation. Now, I shouldn't say concern. My question is, 8 weeks is pretty short as well. So I think it'll be very interesting, even if they see control for 8 weeks. I don't think that gives you any inclination for how long they may be able to maintain that. They are looking for sort of a quick trigger to reinstitute, or to institute, I shouldn't say reinstitute, to institute combination ART. So I think, but it'll be really interesting to see in clinical trial the answer to that question. I think their goal is, and again, the less than or equal to 45 milligram per kilogram gross maintains levels significantly much higher than what's neutralizing. Where I think you may see escape or you may see rebound is in the 5 milligram per kilogram group, because that does fall off sooner. And even though it's above neutralization at 8 weeks, it's not way above neutralization, meaning that, just with the variability in people's virus in people, you may actually see some rebound in that lower dose, which may give them, some information in terms of what the long-term prospects are.
And do you think there'll be differences in the response in people who are newly infected versus people who had been treated and are off treatment for some reason?
I think there may be one for a couple of reasons. One, people who have never been treated and you'll notice that the criteria are 1,000 to 100,000 copies per ml. So that's a relatively low bar. If I can frame it that way, it's a relatively low bar. People who have been on treatment, one, they may not remember or know exactly what all of their different treatment regimens have been. They should remember if they've had any of the exclusionary medications. But, there's a lot more mutations in those people in theory. So I think that they may be, it'll be interesting to see, but I think it may be a higher bar to achieve.
Yeah, I would agree. I mean, one of the things that we've seen recently in sort of looking at viruses in the reservoir, people who start during chronic infection, is that the number of envelope variants is actually astonishingly high in a single person. It can be thousands of distinct variants. And, when people test for bNAb breadth, they're testing, one virus from, 200 different people. But in each of those people, there may be thousands of different variants.
Yeah.
So now it's, of course, less of a problem in people who are treated very early and don't have that much diversity.
Which I think is why that's the group that they're they're trying to get for these very early studies. And, as somebody who works a lot with vaccines, I think this is one of my, I don't want to say issues, but, when you're looking at monoclonal antibody, you're looking at specific, very small targets, right? Single targets with these neutralizing antibodies, which makes escape a lot easier. Now, this, in theory, should be more difficult because it's binding two different sites. But still, and even in terms of vaccine, it's what a comment I wanted to make. If you're trying to make a vaccine with this, you have such a small target, such a small epitope, per se. Trying to get enough of immune response or enough antibodies against that very small target, I think, can be difficult.
Yeah, if I could just ask one more thing. So conceptually, I mean, I agree with you, but the issue is when the antibody's binding to these two different sites, is either site sufficient? Because
Right.
if both binding at both sites is required, then it would seem that it's actually easier to get escape.
I
Because
think, and I should go.
at either one.
Yeah, I think I can go back. I think binding at both sites is required for escape. From what I read, from what I read in the paper, I didn't highlight that on the slide, but I believe both sites are required for escape.
So
Thank you.
it's sort of like a combination in one.
Correct. Yeah.
Yeah.
Yeah. Yeah.
David?
Based on the monkey toxicology studies, what are the expected toxicologies, the side effects?
So in the work that they've done, it's mostly, and I think I breezed by that in my slide as well, is infusion reaction and very few side effects, if any, from the monoclonal antibody, from the antibody itself. It's the infusion of the antibody. So redness, swelling, infection. But in terms of toxicity from the antibody itself, really nothing.
And David Griffith?
Yeah, thanks. Thinking about a patient, I saw a new patient last week who had, I think, acute HIV and had maybe a transient drop in the CD4 count. Do you want to hear about those patients, even though they may have a CD4 less than 400, if it's a transient, kind of in the setting of acute HIV?
Yes, because I think what we would do is we would simply rescreen them and see if their CD4 count comes up.
As long as they stay off therapy.
Yeah,
Yeah.
exactly. Yep.
So
I'll ask him if he started his meds.
Right, so it's a small needle, right? Because it's somebody who's invested enough that they're going to come to a lot of visits, right?
Yep.
But who's also got the equanimity to wait a while before starting treatment. So
Yeah.
it's like but I mean, there are people who fall into that category. But I think one other thing when David, you raised your hand, I was wondering if you were thinking the same thing. Like, this seems like a really good post-birth prophylactic for prevention of vertical transmission. Just wondering if it also, because at the mgs per kg, there's not so many kgs in
Right.
like infants. So like scalability becomes,
Yep.
I don't know, have they been thinking about that?
They have not let us know. I will say this is, I don't know if I can say, they are very tight-lipped about
Okay.
everything.
But that would just feel like, I mean, that's something that Debbie Prasad has brought up about
Yeah.
bNAbs for like 10, I mean, I don't know about 10 years, but remember Mexico City, which is like 6 or 7 years ago. I mean, for something that's dosed milligrams per kilogram, like the scalability in children can be quite high, and infants in particular.
Yeah.
And especially for the challenges around, pill administration and all those sorts of things in that early period of life, it could really provide a period of protection that would be quite.
Absolutely. Absolutely. Absolutely.
Other thoughts or questions?
I just want to thank you for having us here today. And Stuart, thank you for the comment and talking about that. I find it really, really interesting. And I will say, bNAbs, I think, are coming online, not just for HIV, but for many, 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 many different illnesses. They have their advantages. They also have their limitations, but I think it's really I'm very excited to be a part of this study and to work with Joyce and the Bartlett folks and David, you in the ER as well. I'm really excited.
I'll just ask, we have a couple minutes, and I'm curious. So one thing that's generally ignored in antibody therapies is allotypes. Allotypes are allelic variants that groups of people share, but of course that are different between groups of people. I have little doubt that the bNAbs that are developed and the therapeutic antibodies that are developed are largely majority allotypes. None of us knows our allotype, and I
Right.
don't think the drugs are ever indicating what allotype they are. But I can imagine that the half-life of the drug and the reactogenicity of the drugs might vary based on allotype, and I think it's being ignored.
Yeah.
And I think it's a mistake, and it probably disadvantages minoritized people. So, have you ever seen any attention to allotypes in any of the conversations about therapeutic antibody use?
No,
That's what I thought.
no. Yeah.
And I don't think the drug companies want to talk about this because it would mean worrying about this, and they don't
Right.
want it on the FDA's radar.
Right. Right. Yeah.
Interesting. And Ethel, thanks for that link.
Yes, Ethel is linking to a study done of VRCO1 for prevention in HIV-exposed newborn infants. So hopefully the company is secretly considering this
They
antibody
were
too.
described to me by Helen McShane as pathologically secretive.
Well, it's a competitive space, right?
I know.
Yeah.
Well, and disclosure isn't something that the FDA generally rewards companies for.
Right. Right.
Yeah.
Right.
So it's understandable.
Well, thank you. So anybody who has someone who's been diagnosed within the last 12 months is not currently on therapy, but is engaged and interested in learning more. I will say also, I hope they will do some immunology on this because, depending on how close you are to the time of infection, administration of an antibody might either promote or blunt the development of the endogenous immune response, which
Yeah.
could be really interesting to see.
I hope they do. Mary Maravich is with them now, so she certainly should be in tune to that from her work with DAIDS. But yeah, we're collecting samples. They haven't told us what they're
doing with them.
But yeah, they're being collected.
Well, thank you so much, Anna, for coming today. The CFAR hotline number is in the chat, and everyone go free this weekend to experiment with peptides as per recommendations of Health Secretary, and have a great weekend.
You too. Thanks. Bye-bye.